my pleasure to welcome now Georges Lacaud from Cancer Research UK Manchester Institute, a former Patterson Institute, and he's going to talk us about RUNX1 from a developmental biology perspective, knowing that it's a very important gene in cancer.
Thank you very much for the invitation to talk and to share some of our data today. So my lab in the Chaire UK Manchester Institute is interested in blood cell development. And why? Because it's a basic concept, basic knowledge. How do we form blood lineage and cell specification? But also because a lot of the genes implicated in this process, and this is something which we will discuss a lot today, are involved in leukemia, in translocation. And perhaps the best example for this is this gene AML1 for acute myeloid leukemia 1, or RUNX1, which is a master regulator of blood cells development, but it's also one of the genes found the most often translocated either in lymphoid leukemia or in myeloid leukemia. And also what we expect to get for this knowledge from these studies is to understand this program, and if we understand this program, perhaps we're able to recapitulate this program in vitro and to generate cell population by differentiation of ES cells or by reprogramming, which can be used in cell replacement therapy such as bone marrow transfer. So, the first part of my talk will be about the role of RUNX1 in blood cell development. And before to start this, I need to give you a little bit of background about blood cell development. And the most recent data indicate that it's a three-wave system where the first, you don't generate directly hematopoietic stem cells, but you generate more mature cells because it's what the embryo needs. And the first mature cells are generated in the yolk sac, and they are primitive erythroid cells because it's what the embryo needs to develop. Later on, again, still in the yolk sac, you have generation of this EMP, or erythroid myeloid progenitor, and some lymphoid cells. And it's only by E10.5, between E10.5, 12, and 11.5 that the first hematopoietic stem cells are generated. So most of the early life of the embryo, if you want, is secured by these primitive erythroid and EMP cells, and hematopoietic stem cells came only after. So if we look at the cellular origin of these cells, so if we look really early, what has been shown is that there is generation of this blood island, which consists of blood cells, red blood cells, surrounded by a layer of endothelial cells. And this has led to the hypothesis that these cells are generated by common precursor, which has been named an hemangioblast. And this has been postulated more than 100 years ago based on histological observation, and we can define this hemangioblast as a non-differentiated mesodermal precursor that can give rise to endothelial and hematopoietic precursor. If we go later in ontogeny, and mostly in this AGM region or in the dorsal aorta, what has been observed is this emergence of cluster of hematopoietic cells, which are emerging from the lining of this dorsal aorta. And this, the precursor from these cells, has been postulated to be a hemogenic endothelium, which gives rise to hematopoiesis. So an endothelium, we can define this as mature endothelial cells that generate hematopoietic cells. So there is this transition in the dorsal aorta. So there is these two concepts of developmental hematopoiesis, hemangioblast, hemogenic endothelium, and in general they oppose. People I think one or the other. And why is it so difficult to really work on this, to understand what's going on? Is because if you want to work in this embryo, you're really limited by the amount of cells you can access, and it's difficult to study this. So what we've been using in the lab is embryonic stem cells as a model, and the beauty of this model is that it provides access to large number of cells at this early stage of differentiation. And we go with, so most of the work is done with murine ES cells. So we go with cells in presence of a leaf on murine embryonic fibroblasts. To differentiate them, we remove fibroblasts and we differentiate them as embryonic body. And really the trick to use this system is to have a very good synchronous wave of differentiation. And this is shown on this gene expression panel from day 0 to day 6, and you can see that you have a lot of the genes which are expressed in epiblast, mesoderm, and after early hematopoietic development, you can see sequential expression of these genes. So it's a beautiful system because all you have to do is to open your incubator and get these cells and study what's happening in these cells. And in fact, this system has been instrumental to demonstrate the existence of the hemangioblast. Because if you take these embryonic bodies between day 2.5 and day 4, you dissociate them, you replace them in presence of VEGF. What you get is this typical blast colony. These are clonal colony because they're generated in methylcellulose, and you can show that these colony contain endothelial and hematopoietic progenitors. So they seems to be what we expect from an hemangioblast. So cells which are mesodermal cells which can give rise to endothelial and hematopoietic cells, and these cells express the brachyury mesodermal factor. Later, this precursor was also shown to exist in the embryo, in the yolk sac. You can purify these cells as a brachyury FLIC1 positive, so FLIC1 is a receptor for VEGF. If you get these cells replate, you get this colony, this blast colony forming cells, which are the equivalent of the hemangioblast. Okay, so this was one step. So what we wanted to address, the question we wanted to address in the first study I want to discuss today is how do hematopoietic cells are generating from the hemangioblast? Because we know we get this hemangioblast, we replate after 4 days with this blast colony, which contain hematopoietic precursor, but which we don't know what's happening with these 4 days. And again, this is difficult to study in vivo, but we have the in vitro system where we can study this. And to make a long story short, what we did is to follow these 4 days, to do time-lapse imaging, to do a lot of facts. And to make a long story short, what we found this ball of hematopoietic cells is not generated as cells proliferating, but first you have formation of a cluster of tightly associated cells. And to summarize this data, what we were able to show is that this cluster of tightly associated cells are endothelial cells. So in this, so what we're saying in this study is that the hemangioblast generates hematopoietic progenitor through an hemogenic endothelium. And we're putting together these two concepts of developmental hematopoiesis. And also what we've shown in this study that I'm not showing you here is that RANX1 is critical in this transition between hemogenic endothelium and hematopoietic progenitor. If we don't have RANX1, these cells are stuck, and we don't have this endothelial to hematopoietic transition. So we were lucky enough to publish this with some of our colleagues, which came up with the same ideas. So this paper showed that lineage tracing with a VCAD that all hematopoietic cells are generating from endothelial cells. This is a paper by Tim Schroeder. We've done single cells imaging with the ES system and really show that it's not asymmetric cell division. It's really an endothelial cells becoming an hematopoietic cells. In this paper from Nancy Speck, what they've shown is that RANX1, as we've shown with the ES system, RANX1 is critical for this endothelial to hematopoietic transition. And in this free paper, which came 1 year later, what they've shown is that you can, by live imaging, you can show this transition from endothelial to hematopoietic either in the zebrafish or ex vivo in the mouse embryo. So based on this, what we've done in the last few years, we were part of a consortium of 5 groups, which include David Wester, Valerie Kuzkov, Constance Bonifer, and Bertie Goetens. And what we've done is we take all these sequential stage and look at dynamic of gene expression, dynamic at chromatin. And this has been published now as a resource. If you're interested, you can look at it. We know which transcription factor are perhaps the most important at different stages of this hematopoietic development, and also this study has shown that EPO signaling pathway is important for this hematopoietic development, and this was not shown before. What we really interested in my lab was to identify what are the transcriptional targets of RANKS1, which are important for this transition from hemogenic endothelium to the hematopoietic cells. And to do this, what we've done like all of us, we've done the equivalent of ChIP-seq, DAM-ID, and we've done the RNA-seq between wild type and knockout, and we asked what are the genes which are differentially expressed in absence of RANKS1. And also what I should mention, we did really this at the onset. And why are we using DAM-ID? What is DAM-ID? So this is an alternative to ChIP-seq, which allow you to work with less cells, and you don't need to have a very good antibody. When we did this, it was not really the case to have good antibody, a cheap antibody grade for RANKS1. And this has been developed by the lab of Bas van Stinsel at the NKI. And the idea, you fuse your transcription factor with DAM, DNA adenosine methylase. This, when the transcription factor binds, it will methylate GATC. Closed site, this activity do not exist in eukaryotic cells. And what you can do, you just harvest your cells, you digest with DPN1, you amplify, and you sequence to know where your transcription factor is binding. So when we did this, so what was reassuring, we were able to find binding of RANKS1 to non-site of non-target of RANKS1, so SOC17, of this gene AI467, and also in GFI1. And to make a long story short, when we put this all together, what was really striking, we were expecting to see a lot of hematopoietic genes, but what we see is mostly when we do ontogeny is cellular movement and migration. So indicating that RANKS1, the first thing that RANKS1 is doing, it's not really to inducing an hematopoietic program, but probably it's implicated in this hemogenic endothelium, in the aggregation of this hemogenic endothelium, and perhaps the specification of these cells. So this was the main conclusion of this, is that RANKS1 positively regulates both an hematopoietic and cell adhesion program in hemogenic endothelium. And perhaps this has some application because now there is data coming, emerging data suggesting that RANKS1 is mutated in solid tumor, such as breast tumor or prostate tumor, and perhaps RANKS1, this activity of RANKS1 may be important in the potential of these cells to metastasize. So, so this was really a global analysis. What we did also, it's a more specific candidate approach in which we did, again, a differential expression between RANKS1 plus minus and minus minus. We got quite a lot of gene differentially expressed. So this is blast cells development day 1, day 2, day 3, day 4, plus minus versus minus RANKS1. And what we, at this stage, you have to decide which genes you want to study. And we decided to go for 2 genes, which are GFI1 and GFI1b, that I will show you later, because they're transcriptional repressor. And the idea for us is perhaps when you want to move in development, perhaps you want to open new door, but perhaps you want to close some doors. So it was interesting to see what was the role of this transcriptional repressor in this developmental program. And this is the expression of these 2 genes, so you can see you have much less expression in absence of RANKS1 than in presence of RANKS1. And what are they? So they've been mentioned already, some of these genes. So they are zinc finger repressor. They are functionally, what is important for what will come after is they've been shown they're very homolog in the SNAG domain and in the zinc finger domain, and they've been shown to be functionally interchangeable. The SNAG domain, so this is a DNA binding domain, and the SNAG domain stands for Snell GFI1. So perhaps there is something about metastase and EMT here. So there is quite a lot of knowledge about these genes. So GFI1 knockout has been shown to be very important for neutrophil development, and it's also a gene which is implicated in hematopoietic stem cells renewal. And GFI1b is an essential transcriptional regulator of erythroid and megakaryocyte development. And in absence of these genes, it's embryonic lethal. These mice are born and are okay, but these mice die during gestation at E15, E14.5, E15. And if we put this on the hematopoietic tree, GFI1 is important in neutrophil development, is important in hematopoietic stem cell self-renewal, and GFI1b are very important for red blood cells and megakaryocyte development. So the first question we wanted to ask, so we have ChIP-seq data also to show that the direct target of RANKS1, and the idea was, okay, the target of RANKS1, but are they able to rescue some of the phenotype we have in absence of RANKS1? So to do this, we generated a RANKS -ES cells in which we have an inducible DOX GFI1 or GFI1b cassette. And with this, we can ask, we know this is blocked in absence of RANKS1. We can ask if we take this hemogenic endotherm, which expresses type 2, this endotherm marker expresses GFI1, do we get this change in morphology? So the cells are coming from, are changing from adherent cells to loose round cells. Do we get upregulation of hematopoietic marker, downregulation of endothelial marker? And are these cells, if we get these round cells, are we committed blood cells? And indeed, when we re-express GFI or GFI1b, we have this change in morphology. We can see that we have much more of this one colony. We can show this also by time-lapse imaging. So this is when we express GFI1. You can see that this cluster of endothelial cells will round up. If you don't express, this is what we expect from RANKS minus hemogenic endotherm. We don't have formation of viable round cells, so they just stay flat. The next question, are they upregulating hematopoietic marker and downregulated endothelial marker? So our best hematopoietic marker, early hematopoietic marker is CD41, alpha-2b integrin, and our endothelial marker is V-cadherin, cadherin 5. And if we don't induce doxycycline, these cells stay as V-cadherin and do not upregulate CD41. If we induce GFI1, a lot of these cells upregulate CD41 and a lot downregulate the endothelial marker. So they're really doing this endothelial to hematopoietic transition. The last parameter we ask is, okay, we're getting these round cells, they upregulate hematopoietic marker, downregulate endothelial marker. Are these round cells able to form hematopoietic colony? And in fact, they're not able. So they're not committed, really fully committed to blood cells. So what I've shown you so far is the expression of GFI or GFI1b in RANKS minus ES cells, recapitulate or rescue some of the expected change during the EHT. So we get a change in morphology, loss of endothelial marker, characteristic of the EHT, but we don't have complete hematopoietic commitment. Now, the problem we had with this at this stage is that we know that RANKS minus embryo die at 12.5. GFI1 knockout is not embryonic lethal. GFI1b is embryonic lethal at E14.5. So if we say they're so important downstream of RANKS1, why are we not getting a more severe and earlier lethality? So to try to address this, what we have to hypothesis is that there is some functional compensation. And to test this, we got from Tariq Maury two knocking mice in which he knocking GFP in the GFI1 or in the GFI1b locus. And what we can do, we can cross these mice to get double knockout. And when we do this, we expect 1 out of 16 of this embryo to be double knockout. So we get this embryo up to E11.5, E12.5. But when we look at this embryo, we can see that there is a lot of cells which are dead. So it seems we recapitulate more or less what's happening in RANKS minus. Another striking observation was that if you look in normal embryo, so I told you, so GFI1 and GFI1b mark all hematopoietic cells with the GFP, and in normal embryo, these cells are generated in the yolk sac, and as soon as the heart beats, they will migrate in the embryo. if we do this in this double knockout, what we see, a lot of these cells are stuck in the yolk sac. We see almost no GFP-positive cells in the embryo. And these cells still express endothelial marker. And this is just confocal imaging of this, showing you these GFP-positive cells in this Et embryo. And if we look in this double knockout embryo, you can see that these cells are stuck in the yolk sac, and we get almost no cells in the embryo. So we can explain this by different way to interpret this observation. Is that perhaps there is a vascular defect in this double knockout, and we did CD31 staining, and we don't see vascular defect. The other possibility was that there is an absence of heartbeat in this embryo, and it's not the case because we can take this embryo, this is the same embryo, you can see these cells are stuck, but we have heartbeat of this embryo when we dissect this embryo. So the last possibility is that in fact these cells are not liberated in the circulation because they maintain endothelial molecule. And a lot of these endothelial markers are cell adhesion molecules. So endothelial cells needs to be really stuck together. And perhaps because we don't regulate this marker, these cells cannot migrate. So, okay, these cells cannot migrate. They're stuck in the yolk sac, but are they competent blood cells? Did they become competent blood cells? And now what we can do, we can take this 9.5 yolk sac, we can trypsinize these cells and replace them in blood cells assay. And when we do this, indeed, we're able to show that these cells can generate blood cells colony. Of course, we're not getting megakaryocyte and erythrocyte in this colony because we know there is defect in development of these cells, but we're getting haematopoietic colony. And what is striking, even after the formation of the colony, these cells are still expressing endothelial markers such as V-cadherin and Ty2. This is Et cells, and we can show this by FARC, so we can show this by PCR. But these cells are blood cells because they express haematopoietic transcription factor. So if I put this all together, what I've shown you is that downstream of RANX1, GFI1 and GFI1B are very critical to downregulate the endothelial program, but it's not enough to induce the haematopoietic program. And conversely, if we delete GFI and GFI1B, these cells are generated, but they cannot be liberated and cannot be disseminated in the embryo. But they are blood cells, committed to blood cells fate. So this was mostly done with the yolk sac or with the ES cells. So we wanted, because we have this EST happening in the yolk sac or the EST happening in the AGM, we wanted to ask, is it the same thing happening in the embryo during generation of the haematopoietic stem cells? Because this is perhaps a more important question. And to do this, what we did, staining of this dorsal aorta, and we generate our own GFI1 H2B tomato, so we can distinguish GFI1 from GFI1B. So we have two reporter H2B tomato for GFI1, GFP for GFI1B. And when we cross this, we can see that this cluster, which are c-KIT positive cells, GFI1 seems to be more at the basis of this cluster, whereas GFI1B seems to be more when these clusters are emerging in the dorsal aorta. And this is recapitulated on this cartoon. We think that this is the endothelial cells, endothelial lining. We can now identify hemogenic endotherm. We don't have marker for hemogenic endotherm because these cells express GFI1, as these cells will after start to bud off of the endothelial lining, upregulate c-KIT, and after they will upregulate GFI1B. So we think now we can prospectively isolate these cells. So this is what we've done. And indeed, when we take these cells and do, this was done with vertiglutans, and this is old 3'96 gene, endothelial or haematopoietic genes. And when you look at expression of these genes in this different population, V-cadherin, or this V-cadherin GFI1 upregulating c-KIT and upregulating GFI1B, you can see that it's a continuum. It's really these cells are able to generate these cells. And also we purify these cells and show that they can generate the next population. And GFI1 seems to be more associated with endothelial genes, and GFI1b is more associated with hematopoietic genes. So again, this is consistent with this progression. And again, this population, we can see a lot of these endothelial markers are downregulated, and a lot of these hematopoietic markers are upregulated in this different population. The next question was, is it really these cells which generate hematopoietic stem cells? And to answer this question, we purify this V-cadherin expressing GFI1 and V-cadherin not expressing GFI1. We did transplantation, and we can show that indeed these cells which express GFI1 are the ones which will repopulate mice. We've done it with GFI1b also, and they're more potent as the cells mature, they're more potent to repopulate mice. So, but indicating that we're really looking at the cells which will generate hematopoietic stem cells. So, the next question, are these two genes important for this transition, again, in the dorsal aorta? And I'm just showing you the double knockout and the 8-8. And you can see emergence of this cluster in the 8-8 mice. And you can see that this is completely altered in the double knockout. So the cells stay in the lining of the dorsal aorta and cannot emerge, cannot do this hematopoietic to endothelial to hematopoietic transition, cannot form this cluster. So really what we can conclude from this is that GFI1 mark hemolytic endothelium, which gives rise to hematopoietic stem cells. So the next question is, what are these two transcriptional repressors doing in this, during this transition? And from this, we get some clues from a study of Stuart Orkin, which has studied the role of GFI1 and GFI1b in EML cells during differentiation. And what they've shown in this paper is that GFI1 and GFI1b recruit the correct repressor complex and HDAC. These take out positive mark of transcription, and G9A, we completely silence this locus, this erythroid locus. And what we have in this complex is LSD1, this histone demethylase, HDAC1, HDAC2, this histone deacetylase. So we wanted to ask, is the LSD1 also a major player in our hematopoietic endothelial transition? And from this, we took advantage. One of the group was working on a LSD1 inhibitor in the institute, and we got some of this inhibitor. And when we do this Blaskoni differentiation, you can see if we add this inhibitor, we completely block this transition from this cluster of cells to these swarm cells. And it seems it's directly correlated. You have more of this and less of this. We did this also during this Blaskoni culture. And this is FAX. This is with the LSD inhibitor. This is with the MSO. You can see that in the MSO, these cells, these endothelial cells, upregulate CD41 and downregulate Tie2 or V-cadherin, these two endothelial markers. If you do this in presence of this inhibitor, you completely block this transition. These cells cannot downregulate their endothelial marker. So this was done with a drug. So we were a little bit afraid to base everything on a drug. So we were lucky enough to get from Sean Conley at the University of Leicester an inducible LSD1. And this was important because LSD1 is required for mesoderm development. So we really induce the deletion when we do this Blaskoni assay. This is what happens if you don't delete it. You see this transition, the endothelial cells upregulating CD41 and downregulate endothelial marker. If you do this with this inducing the deletion of LSD1, you block this transition. So really indicating that LSD1 is required for this transition. So what are the genes targeted by this? So to do this, we did RNA-seq between the different the cells progressing or the cells block. We did also DAM-ID for GFI and GFI1b binding. And if we put all this together, what we get a restricted set of genes. But was really striking, a lot of these genes are endothelial genes. And also, a lot of these genes have RANX1 binding, and a lot of genes have also Notch1 binding. which is, we know it's an important player also in this endothelial to hematopoietic transition. So really what we think here is we're showing directly that GFI1 is important to downregulate the endothelial program. So if we put this all together, this is a RANX1-centric view of hematopoietic development. We think that RANX1 in this hemogenic endothelium, first RANX1 is perhaps implicated in the adhesion of this hemogenic endothelium, of this specification of this hemogenic endothelium. But during this transition, RANX1 induces the expression of GFI and GFI1b, which will be recruited at the level of endothelial genes. We think perhaps RANX1 is already binding here because we see a lot of RANX1 binding. So perhaps there is a cooperation between the two genes. This will recruit the co-rest repressive complex, and this will shut down this endothelial program in this hemogenic endothelium. But we know that if we express GFI and GFI1b, this is not enough to get hematopoietic commitment. So what we have to include in this model is that RANX1 on its own is also inducing the hematopoietic program, an hematopoietic program. So really, during this transition, you need to downregulate an endothelial program and activate an hematopoietic program. And we don't know really what are the most critical genes in this transition, and we're working on this. So really where this leads us is that now we can prospectively, for the first time, isolate hemogenic endothelium and study the transcriptional program, which leads to the development of hematopoietic stem cells. And this is what we've done and we're still doing in getting this population and doing single cells, RNA-seq. So we've done this in collaboration with Catherine Robant and Van Wijngaarden at the Utrecht Institute, but more recently we've done it in our own lab. And the idea is that we can isolate this population, we can put them on a pseudotime, we can look at expression of genes. So GFI1b, for example, will be something we expect at the end. We validate this. We've done this by SMART6, so we can look at transcript during this transition. And also we can use this to try to find new players during this transition. And these are some of two new players we found during this transition. So V-cadering is downregulated, RANX1 is upregulated. And we found that Icaros and Helios are really expressed during this transition. And we can validate this at the expression level, because here we're standing for Helios, and we can see really Helios is really nicely expressed in this cluster. And we're doing more study to identify new genes and also to identify, to functionally characterize these genes using the CRISPR technology in embryo. So this is where we stand in our work, in your study, trying to understand developmental hematopoiesis. Now, something I should mention also, what we're trying to do, we're trying to use this RNA single cells, RNA-seq, to ask the question, what is the difference between an hemogenic endothelium in the yolk sac, which generate EMP cells, and a hemogenic endothelium in the AGM, which generate hematopoietic stem cells, with the idea to understand what should we add to embryonic stem cells differentiation, because they mostly recapitulate yolk sac hematopoiesis to allow these cells to generate hematopoietic stem cells. And this fits really well with what our talk to now it's the role of RANX1 in reprogramming. So and the idea is, can we reprogram fibroblasts to blood cells? And as you know from the work of IPS, we can reprogram this, but also the work of Weintraub, but MyoD can reprogram fibroblasts to muscle cells. But there is also example inside the hematopoietic system that you can change cell fate with transcription factor. So we wanted to do this. At the stage when we started this, there was this publication which excited, I guess, a lot of people in the field, from Miki Bhatia, which show that if you express in human normal fibroblasts OCT4 and put these cells in the right condition, you can get a lot of blood cells. So the idea was, can we use this? Can we understand how this work? And we tried to do this, a lot of labs tried to reproduce this, and it didn't work in our hands. So we had to go after something else. And what we decided to do is to, like Yamanaka has done, is to take a bunch of hematopoietic transcription factor, early hematopoietic transcription factor, and ask if we express them in fibroblasts, do we get blood cells. And the methodology was the following, is to get murine embryonic fibroblasts from embryo or adult fibroblasts to really get rid of all the endothelial and hematopoietic cells in these fibroblasts, to bombard them with transcription factor, with retrovirus expressing transcription factor, and to screen for the presence of one cells colony, and to assay if these one cells colony are hematopoietic cells. And indeed, this work. We were able to get this one cells colony. This is the 5 transcription factor which works the most robustly in our hands, so RANX1, SCL, LMO2, GATA2, and ERG. We can, this is we've done with embryonic fibroblasts, adult fibroblasts. Each dot is a different experiment, so you can see that it's relatively reproducible. It's quite reproducible, but it's not, the efficiency is not really high. If we get these one cells, we can generate hematopoietic colony, and we can get myeloid, erythroid cells. We have some indication that we can get some B and lymphoid cells, so we can get here some B220 CD19, and we can show some BCR rearrangement in these cells. We can do this for T cells using OP9 co-culture. We get this CD25, and we can see some TCR rearrangement. I'm not saying that they're fully mature, they're really bona fide lymphoid cells, but they're on their way to become lymphoid cells. So this work, another question was, how do this work? Are we going directly from this fibroblast? Are we going directly to a differentiated or hematopoietic progenitor, or are we going through ES cells? Are we recapitulate just ES cells differentiation, or through an hemangioblast or an hemogenic endothelum? And we look for ES cells marker, OCT4, nano, we didn't see any. Hemangioblast, we look for brachyury, we didn't see any. We look for endothelial marker, and indeed, we take this transition, we harvest the cells at 3 weeks. We can see them before the colony of one cells. But if you look during this 3 weeks, you have really a peak of endothelial marker, suggesting that we recapitulating what's happened during ontogeny of the hematopoietic system. We're going through an hemogenic endothelium. And to test this, to validate this, what we've done is to purify these weak adhering positive cells, the first cells emerging at day 6. We put them on OP9 to allow them to mature, and we replate them, and we show that they're able to generate hematopoietic colony, showing that really these cells are hemogenic endothelium. And we can show this also here, where we are doing NS-seq on this weak adhering and later c-KIT hematopoietic progenitor, and you can see these cells express endothelial marker, but they're already starting to express some of these hematopoietic markers, so they're really on their way to generate hematopoietic cells. So the major conclusion of this is that hematopoietic transcription factor could reprogram fibroblasts to blood cells. We have multi-lineage ability of these cells, so we can take individual colony, they have multi-different lineage, hematopoietic lineage. We've shown that this reprogramming occurs through an hemogenic endothelium stage, and what I haven't shown you is that we have repopulation, but a really short-term repopulation, so we don't have bona fide hematopoietic stem cells. So we're still working on this. So the idea will be, can we improve this in vivo engraftment? And to do this, we base this on the work we've done with Valery Kruskov, where we've shown that really the first c-KIT cells which emerged during this differentiation are the cells which give the best repopulation. So we got these c-KIT protein cells, and we put them like Shahin Rafi has done. We put them on a vascular, on endothelial cells. And if we do this, we can get in some experiment long-term repopulation. But this is still something which is not really robust. We don't get it anytime, so we haven't published this yet. We're still working to improve this. So the next question was, can we reprogram human cells? And indeed, we're able to do this, so we can take this human dermal fibroblast, we express with 5 transcription factor, we get this CD45 positive cells, and they have morphology of hematopoietic cells. What we're trying to do now is to understand better what is the transcriptome change during this reprogramming. Also, we would like, because it's not something which is very efficient, so we would like to screen for a factor or compound which will allow us to improve the efficiency of this reprogramming. And with this, I will stop here. So I think I mentioned some of the people doing this work are founding and our long-term collaborators, so Valerie Kuskov, Constance Bonifaire, Bertie Goethens, Catherine Robin, Sean Conley, and Tariq Moroy for the GFI 1B mice. Thank you very much.